Muscle cells are the body's specialists in movement. They contract linearly using highly organized arrays of actin and myosin filaments. This generates force in one direction, like pulling a rope. This is effective for large-scale movements. However, non-muscle cells move in a more subtle and intricate way. Non-muscle motility is essential for life. It underpins processes like cell division, immune responses, and wound healing. Unlike muscle cells, non-muscle cells don't rely on linear contraction. Instead, they use dynamic and localized changes in their actin cytoskeleton. This allows them to crawl, change shape, and interact with their environment in more complex ways. The key to understanding non-muscle motility lies in the cell cortex. This thin layer beneath the plasma membrane is rich in actin filaments. These filaments are not static. They constantly assemble and disassemble, creating forces that push and pull the cell membrane. The regulation of actin dynamics in non-muscle cells is complex. It involves a diverse group of proteins that interact with actin filaments. These actin-binding proteins control filament assembly, disassembly, and organization, orchestrating the intricate dance of non-muscle motility. The cell cortex is a dynamic region just beneath the plasma membrane. It plays a crucial role in non-muscle motility. This thin layer is primarily composed of a dense network of actin filaments. These filaments are not arranged in the rigid repeating units seen in muscle cells. Instead, they form a more fluid and adaptable meshwork. This dynamic network of actin filaments in the cell cortex is constantly changing. New filaments are assembled and added to the network, while others are disassembled and removed. This constant turnover of actin filaments allows the cell to rapidly adjust its shape and generate forces. These forces can push the cell membrane outward, creating protrusions like lamellipodia and filopodia. These protrusions are essential for cell migration, allowing the cell to explore its surroundings and move towards a target. The cell cortex also plays a crucial role in other forms of non-muscle motility. For example, during cytokinesis, the process of cell division, the cell cortex contracts to pinch the cell in two. The dynamic nature of the actin cortex is essential for its function. This dynamic behavior is tightly regulated by a diverse group of proteins known as actin-binding proteins. These proteins dictate how, when, and where actin filaments assemble and disassemble, orchestrating the intricate movements of the cell. Actin-binding proteins, or ABPs, are the master regulators of the actin cytoskeleton. They determine the behavior of actin filaments, influencing their formation, breakdown, and organization. This intricate control is crucial for non-muscle motility. Just like a choreographer guides dancers, ABPs dictate the intricate movements of actin filaments within the cell. There are various types of ABPs, each with a specific role. Nucleating proteins, like formins and the ARP2/3 complex, initiate the formation of new actin filaments. These proteins act as starting points for filament assembly, promoting the growth of the actin network. Capping proteins, on the other hand, bind to the ends of actin filaments, preventing further growth or shrinkage. Severing proteins, such as cofilin, break down existing filaments. This creates a pool of free actin monomers, available for building new filaments elsewhere in the cell. Cross-linking proteins, like filamin and alpha-actinin, connect actin filaments, creating a more stable and organized network. This intricate interplay between different ABPs allows the cell to precisely control its actin cytoskeleton. By regulating the assembly, disassembly, and organization of actin filaments, ABPs orchestrate the complex movements required for non-muscle motility. Non-muscle motility is essential for a wide range of cellular processes. One striking example is cell migration. During this process, cells extend protrusions at their leading edge, using actin polymerization to push the cell membrane forward. These protrusions then adhere to the surrounding environment, providing anchor points for the cell to pull itself forward. This coordinated dance of actin assembly and disassembly allows cells to move, a process crucial for wound healing, immune responses, and embryonic development. Another example is phagocytosis, where cells engulf large particles like bacteria. During phagocytosis, the cell extends arm-like projections driven by actin polymerization to surround and engulf the target. This process is crucial for immune defense, allowing specialized cells to capture and destroy pathogens. Cytokinesis, the final stage of cell division, also relies on non-muscle motility. During cytokinesis, a contractile ring composed of actin and myosin filaments forms around the middle of the dividing cell. This ring constricts, pinching the cell in two and creating two daughter cells. These examples highlight the importance of non-muscle motility in various biological processes. From cell division to immune responses, the ability of non-muscle cells to move and interact with their environment is essential for life. Understanding the mechanisms underlying non-muscle motility is crucial for unraveling the complexities of cell behavior and developing new therapies for diseases related to cell movement.